70 studies now in the public domain on PRP. It all dates back to Dr. Mishra's first paper published in the American Journal of Sports Medicine in 1998, I think it was. And I don't know how many of you remember how many patients were in the study. Anybody know?
12.
Right. There was 15 people in the study. When you read the abstract, you'll see there's over 150 that he evaluated, but only 15 of them had the therapy, and 5 of them had just Marcaine, and 3 of those 5 were lost to follow-up. So he had no follow-up on the control group. And if you, again, I would encourage you to revisit the paper. An entire $250 million a year industry has been built upon that paper. The only study that ever showed a benefit, randomized prospective study, was the Peerbooms article that was published in Lateral Epicondylitis, American Journal of Sports Medicine, 2006. And that showed, that was a study against steroids. And if you, again, look at the data, if there's any scientists in the room, the first time point after the therapy was at 4 weeks. The patients that had the PRP injection were doing much worse because PRP really, really hurts. And if you remember, before anybody started, see, it's like a cat and mouse game. Once the studies come out to show the stuff doesn't work, the guys that are doing it already switched to something else. So in the beginning, it was always leukocyte rich. We forgot about all that, right? The only PRP that was out was LR PRP. We didn't call it LRP, we just called PRP, but it was all leukocyte rich. And as soon as all the articles came out, one of them was mine. I never even had a product. Published in the JBJS. I would strongly encourage you to look at it, showing that it accelerates degeneration of cartilage, chondrocyte death. They're chondrotoxic. And at a later time point, the PRP patients did do better because it's against steroids. We all know steroids are tenotoxic as well. There's a tendon study. So I'm not a big fan of PRP. We now have leukocyte-poor PRPs. I just came back from the Academy of Orthopedic Surgeons at the end of March, and now they're saying, the new stick in my world in orthopedics is, so we went from any PRP worked, even though if you Googled it, it hurts like hell, is the thing to get. And then they said, okay, you gotta use leukocyte-poor PRP. And now they're saying that it doesn't matter what the concentration of platelets is. So if it doesn't matter what the concentration of platelets is, then it can vary from no platelets to a whole bunch of platelets. Then actually, what's the PRP doing? We've already gotten rid of the red cells, we've gotten rid of the white cells. Now, the new data that's coming out is all, but there's no question that something works. And I would submit to you, if you look at A2M concentrations, and again, go on clinicaltrials.gov, the study that I did, looking at the amount of A2M, and A2M in the blood is that there's a wide variation. If I took everyone's blood here right now, the variation is between 0.1 mg per ml to 1.5 mg per ml. There's a massive difference. And I would submit to you that the higher amount of A2M you have in your blood, when you get rid of all the other stuff and inject it into your joint, those are the patients that probably did the best. Now, all the systems that are PRP concentrating don't concentrate A2M, so whatever amount you have in your blood is the concentration. It's what you're going to get 1x. So systems that concentrate A2M, in my opinion, and again, I'll know in 2 weeks if I'm right, but I'm just not a big believer in PRP because the data doesn't support it. When we, not just anecdotal data, we did this many patients and they all degrade, or 80% of them degrade. Looking at a comparator control of patients with saline injections, because you really can't even compare them to hyaluronic acid or steroids. The FDA won't even allow it anymore. So that's my opinion on that.
I will take a little bit the opposite side of that. I consider PRP as the form of stem cell therapy. And Galliapin is right. Is it really the platelet? Is it the leukocytes or whatever? But if you look at the way people do PRP, it's so variable in the techniques. The centrifugation technique that people use varies from the use of simple centrifuge to industrial centrifuge to isolate. right there is a difference already in the composition of what you inject. And just like SVF, really and truly, PRP is a soup. It contains anti-inflammatory substances, cytokines, stem cell, etc. Therefore, to me, PRP is actually a misnomer. Unfortunately, because of this variability in technique and collection, the results published in the medical literature is variable. True, there are quite a few numbers that show failure, but there are also quite a few numbers that show success. The bottom line is, for those of you who are doing PRP, and if you have success, I cannot argue with that. But in my practice, really, I use mainly PRP as a biologic scaffold to do stem cells. But I know that other people are doing it primarily, you know, in treatment of plantar fasciitis or tendonitis or arthritic flare-ups, etc. And I've seen it work. So I will not actually say that it doesn't totally work. I think there's no uniformity by which the soup is prepared.
Thanks. I'll just very briefly say that when we looked at our centers who were doing our intra-articular injections, we didn't see any difference between the people who routinely use PRP with the SVF and people who don't. So, you know, I think the jury's still out on whether, some people were thinking, well, it's almost like that hamburger helper or something. There's a lot of, you know, fresh cytokines in there or growth factors or something that might improve cell function. But we need to look at this more. There's no evidence. Any other questions?
Where is the A2, if you could?
I would suggest you go so all the other people can hear the questions.
Thank you. Thank you, Oli, for wonderful lectures. Dr. Scuderi, the A2M, is that available? Is there a process to obtain the A2M to utilize in patients?
Yeah, so we got a 510K approval, the Cytonix lab did, for a system that concentrates alpha-2 macroglobulin about 6 times what's in your blood. And we eliminate all the small proteins and the pro-inflammatory cytokines, as well as other important proteins that are in your plasma for other functions unrelated to cartilage restoration. So there are 484 proteins in our plasma that we know about. There was a couple of dozen that were just discovered about a year ago. And probably, if I come back next year, there might be 515, because there's new proteins we discovered all the time in the plasma. And then, of course, we find out what they are. But the system that we built concentrates A2M to a significant degree. It's made by Cytonix Laboratory. And again, the study that we're doing is an FDA-approved study, so we can get an indication for osteoarthritis of the knee, so I can get the therapy to all patients, not just the rich ones.
I have a question. I think it's really interesting to hear this. You know, we don't really know what's in the PRP. Is there a magic bullet? Is it alpha-2 macroglobulin? We don't know. But I think it's important to point out, Dr. Scuderi, the Wang study and those studies were done in animals. There's no human studies that were done in alpha-2 macroglobulin except for the ones that you're doing, which we don't have data. So I don't understand why you would sort of trash the PRP studies, even because we have studies on that. I mean, some of them are not that great, but some of them are. There are human studies that show osteoarthritis is treatable with PRP. Now, I'm not a big fan of anything here. Alpha-2 macroglobulin may be the magic bullet. I think it's also important to point out that alpha-2 macroglobulin is available through your company. You're the CEO of Cytonix. You are the one making this. You are the one that plans to sell it. And I'm fascinated by this, but I've talked to other regenerative gurus in here, and they've said that there's 2 studies in rats, 1 rat and 1 rabbit, that show some decrease of osteoarthritis. So I think PRP, we should give a little bit more benefit of the doubt to that. This whole field is new and exciting, but I also think that saying alpha-2 macroglobulin is the answer for everything. There's just no evidence to support that.
So twofold, you made 2 points there. One, that I'm trashing the PRP studies, and 2, that I'm recommending A2M to be used for everybody. I don't want anybody to use A2M. My goal isn't for you guys to buy kits. My goal is to get FDA approval through the normal process so everyone can get it. The company will be sold if I'm right, and I'll be fine. The research that I've been doing for the last 14 years points very strongly to this mechanism. And you're right, there aren't any studies yet. But I would submit this to you. PRP has been around for at least a decade. How many companies have gone to the FDA with their products and said, hey, we think we have something. My study only cost me $1.3 million. The companies that do PRP, there's 40 of them now. They all have a lot more money than that. I had to scrounge the money to get my study going. That's how confident I am that it will work. But not a single other company has gone to the FDA and done a similar trial. Why is that? And the answer is, there are 70 papers already showing it. And as soon as the study comes out, if my study doesn't work, then guess what? A2M goes away. And I'm okay with that as a scientist, because I'm not trying to make money, I'm trying to help people. But if a company, and you can name your company, of the 40 companies that sell PRP systems, did the same thing and showed that it didn't work, they couldn't sell that product anymore. And it's a $200 million market. And they already know the answer. They're bamboozling you, and they're bamboozling the patients. And to me, that's disingenuous, because it's only $1.3 million, which is pocket change to these people. And the study could have been done already a long time ago. I've had to do this for over 2 years because of funding issues, and no one else has done it. So I would ask, why haven't they just done it and given us the answer of which one is good?
Well, let me answer that. The FDA does not require really testing of PRP because it is an autologous source and it is minimally manipulated. Therefore, it is considered as self. It doesn't have to undergo rigorous FDA testing. It doesn't have to undergo the so-called good clinical practice. I forget the name, but it's the requirement of the FDA in order for the FDA to approve a product. It does not belong to the category. That's why there is no interest in the FDA, by the FDA to regulate something that comes from self. That's the reason why there is no imposition from that regulatory.
Okay, that is absolutely not true. The FDA approves products that are safe through a 510K pathway, and that's the pathway that we're all using for PRP systems. You have a predicate device that's already on the market, and you show that it's not materially different. It's within 20% of specific parameters for a PRP system, the system that I have in the market. You have to do hypertonic shock studies, P-selectin studies, these are platelet aggregation studies. You have to show a concentration of white cells and platelets that are within 20% of the predicate that you're testing. It's a fairly simple process, but what it gives you is no indication. So there's no code that can be used because there's no reimbursement process. In order to get FDA approval, like what I'm going through, for an indication, and the indication that we're going for is osteoarthritis of the knee, you have to do a randomized prospective placebo-controlled trial through the FDA. You have to do tox studies first with a small animal, and then submit your protocol to the FDA for approval. It's a lengthy process. It costs a couple of $1 million, like I mentioned, but in order to get an indication, to get it to all patients, is a very specific process because the FDA is concerned foremost about safety and your label. That's what the FDA approval process is for. It's for a label. 510K is a very different process than what is known as an IND, which is what I'm going for.